Now, I would like to introduce our webinar speaker. Dr. Kirsten Heiss graduated from the Ruprecht-Karl University in Heidelberg in 2006. She completed her PhD in infectious diseases, working on the malaria-causing agent Plasmodium falciparum. This was followed by her postdoc studies at the Institute of Immunology at the University Hospital of Hamburg-Eppendorf. Since May 2011, Dr. Kirsten Heiss is Managing Director of Malva GmbH. So Dr. Heiss will tell us about a way of employing high-density peptide arrays for rational vaccine design in malaria. Dr. Heiss, I'll give the word to you. Please take it away.
All right, thanks for this kind introduction. So welcome to everybody and welcome to today's webinar dealing with peptide arrays and their application in rational vaccine design in malaria. So I will be guiding you through today's webinar in both my capacity as a Malva company representative and malaria scientist who is using the state-of-the-art high-density peptide array technology from PEPperPRINT. I would like to take the opportunity to give you some facts about the Malva company, so about us. So actually we are a young biotech company founded in 2011. We are actually a spin-off of the parasitology unit at the Center for Infectious Diseases based at the University Hospital Heidelberg. And we are particularly focused on the development of a next-generation anti-infective vaccine, which we envisage is composed of novel vaccine candidates recently identified by us, and I will come to that in a few slides. And we would like to use them in a multi-component subunit vaccine, so the MAL vaccine. Actually, the company mainly focuses on research and development and has an interdisciplinary team responsible for the production success and, of course, ultimately the generation of the MAL vaccine. So we are a team composed of, for example, scientists with long-standing expertise in infectious diseases with the focus specifically on Plasmodium vaccinology, but also a clinician experienced in clinical trials, and moreover the company is mentored by, again, a sub-team consisting of advisors with specific expertise such as business development as an example. So actually, so why we are focused on producing a malaria vaccine. And this is summarized in this slide. So first of all, of course, is the question, is a malaria vaccine needed? And the answer is yes, definitely yeah. Is urgently needed, and this is like malaria is the 5th leading cause of death from infectious disease worldwide. And if you have a look on the numbers, I guess it's quite clear. So malaria affects the life of almost 200 million people and causes over 500,000 deaths annually. So actually, especially children under the age of 5 can be seen as one of the most affected group of people suffering from severity induced by malaria blood stage infection. Actually, malaria is caused by a Plasmodium parasite and it's transmitted by Anopheles mosquito. So just briefly, what happens when you get bitten by an affected female mosquito? So actually the parasites are transmitted and the liver, which is indicated here, is the first site for further intracellular development. And this replication phase here in the liver is clinically silent. That means that this replication phase is not accompanied with any disease symptoms. Actually, malaria pathology starts with the transition here of the parasites into the bloodstream, resulting in repeated infection and moreover disruption of the red blood cells. And I guess this is quite common. This is accompanied with the typical malaria symptoms with high fevers and could also possibly lead to death if untreated. To close the cycle, there are also some stages developed in the blood of the human body, which are called gametocytes. So these are sexual stages which are infectious again for the mosquito vector, and then development occurs in the mosquito, and of course your neighborhood could be then be bitten by the next infected mosquito. So we have at the moment no vaccine available, and the problem is also an increasing drug-resistant parasites. So when we look in more detail for the targets for vaccine development, and we are looking again on this complex life cycle of the parasite, so it's indeed that only the phase here in the liver, so the kind of liver stages is the only phase that can be eliminated by sterilizing immune response, thereby inhibiting malaria pathology. So only stopping the parasite here gives you a sterile protection. We should, however, not forget to look at the entire life cycle, where two additional important bottlenecks for the maintenance of malaria infection are crucial. And here we have again the erythrocytic phase, truly the most important one when it comes to disease symptoms. Here a vaccine would minimize the symptoms associated with the Plasmodium infection, and hence an anti-disease vaccine would protect from clinical disease, which is never sterile. So sterile, this we have to keep in mind. A third intervention point here, the prevention of transmission, so of sexual stages to the mosquito vector. This can be disrupted by transmission blocking vaccines. This approach has truly a disadvantage over the two other, as it is often considered as being altruistic. So actually, the Malva company, we are focusing on a strategy to induce complete protection at the stage of the liver, which is shown here. So, of course, anti-infective vaccine development is definitely not new. So since decades, research in malaria is focusing at establishing a technology or a system that allows the perfect composition for a perfect anti-malaria vaccine. So the state of the art, or the two main lines of development, are just summarized briefly in this slide. So we have in malaria vaccine development two approaches. So we have whole organism vaccines, and on the other hand we have subunit vaccines. So common approaches also in other vaccine approaches. So in this whole organism vaccine we have different lines, and what they have in common, they are highly efficacious, so they mediates sterile immunity. But when you look at here at this kind of disadvantages, so in order to get sterile protection, the vaccine has to be injected intravenously, and this multiple times. So at least at the moment you need 5 injections that you end up in a sterile protection. So these are mandatory. And also the production of this whole organism vaccines is quite challenging. So you need the mosquito vector in order to produce the vaccine lines, because you have to isolate the corresponding stage of the malaria parasite out of the mosquito. And of course, since you are dealing here with whole organisms, and they are all kind of attenuated, so there is of course a risk of breakthrough infections while immunization takes place. Actually, on the other hand, you have subunit vaccines. So here you just use an antigen or a part of the parasite, and here antigens from different parasite stages are under development. You clearly see the positive, the advantages. It's a straightforward production, of course, because you have a lot of biotechnological systems to produce the components, and of course it's safe, so it never comes to breakthrough infections or reversions. But a big challenge is that it's so far the current approaches are insufficient in producing or eliciting protection, and also it's a kind of short protection. So we decided to take, for our approach, so for the mal vaccine, the advantage of the two of the both presented lines here and create a novel vaccine line that emulates the existence of a whole organism vaccine with the advantages of a subunit vaccine. So the straightforward production and the safety with the highly efficacious advantage of a whole organism vaccine. So we based our innovation on the existence of these two challenges in malaria vaccine development, but also took in consideration how the immune system would react and should react in response to vaccination. So, and as it is known in malaria immunology, so what is important for protective immune responses, it is known that in order to achieve a sterile protection against Plasmodium infection at this clinically silent liver stages, T cells are mandatory, so T cell mediated immune responses are required. Additionally, it has been shown that antibody-derived mechanisms play also a considerable role in decreasing the parasite infection, which is indicated here, to the liver, and hence contribute to protective immune responses. And furthermore, here at the pathogenic blood stage phase, antibodies or humoral responses are the central effector mechanisms in order to have protection from clinical disease. So the challenge still in vaccine development in malaria is that the entire picture of antigens that are really crucial for inducing this potent protective anti-plasmodial immune responses remains incomplete. So the identification of novel antigens involved in the establishment of protective immune responses will of course open up new perspectives for rational malaria vaccine development. So there are of course certainly several ways to isolate novel antigens, and Malvar has taken a specific approach based on differential expression analysis in order to isolate novel antigens, and this is quickly summarized in this slide here. So we performed an innovative screening strategy based on differential expression analysis, and with that it has become possible to identify critical target antigens from these attenuated parasite lines, the so far only approach conferring sterile protection. So in the next step, these antigens that we identified were validated, and we were able to demonstrate in further studies that indeed T cell responses against a subset of these novel antigens are generated during natural exposure as well as experimental vaccination in mice. And now we have kind of expanded our focus not only on cell-mediated, so on T cell-mediated immunity, but we became additionally curious to also identify humoral responses, at least for a set of our antigens. And in this context of this approach, we then aim to address the question whether or not these novel antigenic determinants are also an essential component for humoral responses during malaria infection or malaria disease. So in order to address the question, we have hence searched for a suitable technology and found PEPperPRINT's high-density peptide array technology as the most suitable one for our application. So in general, what you can think of when it comes to antigen discovery, what you can do, so peptide arrays can be used, of course, as a screening method for a broad screening a broad range of antigens. to identify under this broad range of antigens novel ones involved in antibody-mediated anti-malarial immunity, or more specifically, you can apply such a, I called it now tailor-made approach for the analyzation of a certain set of antigens, and this is exactly what we did with the technology of PEPperPRINT. So we have selected the tailor-made approach to analyze a limited number of our novel plasmodial antigens, but also in combination with already described malaria vaccine candidates. So to summarize it, the general objective is as follows. So it's a down selection of antibody epitopes of our antigen candidates involved in protection from clinical disease and sterile protection. The arrays were designed in a way that we have in total 7 antigens, and these were subdivided in 15 mere peptides. So in total we have 1,104 individual peptides, and they were printed on the arrays in duplicates. We used an overlap of 7 amino acids, and they were printed, as I mentioned, as duplicates on the arrays. And each array is framed by an HA as well as a flag control peptides. The sera that we used and that we are still using and that we will use in the future are kind of summarized here. So we have sera in a collaborative approach from individuals living in malaria endemic areas, differing in age. So the data from this I would like to present today in this webinar. And in a more future perspective, we have sera from individuals from experimental vaccination studies, and these individuals are protected against malaria infection. And as control for both, we have malaria-naive sera. And as mentioned, the technology that we used were the PEPperChip custom peptide microarrays. This slide here just briefly summarizes our experimental procedure, just briefly. So this is what you can see here. This is our incubation tray, so our chamber specific for our array. So we have per glass slide is covered with 3 arrays, individual arrays. But of course this depends on the total number of peptides that are printed on the specific array. So what we do in order to detect positive responses, so we do as a kind of control to look for unspecific binding. We first test with every array charge the secondary antibody to look whether there is an unspecific background. And then we subsequently continue with the incubation of the different sera. This we do overnight. And then we detect the antibodies with a secondary antibody recognizing IgG. And this is coupled to this daylight 680 antibody. Then we do the scanning using a LICO infrared fluorescence scanner. And at the end, we incubate the array slides with the HA antibody to stain for the control peptides, and this is delivered by the PEPperChip staining kit. And then, of course, we have to scan again. So here, what you can see here as well is a kind of analysis workflow, which we can also briefly summarize. So the staining intensity data that we obtained after scanning the glass slides are further transformed for quantitative analysis. So we have our raw fluorescent images that we have after the scan. Then we analyze those images using the PEP slide analyzer software. Then we move on with transformation of the data And then we apply 2 different approaches for the final analysis. So we have a bias regularized t-test, and we also do a cluster analysis, and I would like to show you in a second the first results of our data in a kind of cluster or in a cluster analysis. Before showing you the current results, I would like to go one step back to just give you again a brief overview about the sera that we have used to obtain the data set that I'm going to present. We have in our collaborative approach or project, we have access to well characterized serum samples from individuals living, also malaria exposed individuals, and they are well characterized in a way, in a sense of the clinical point of view. So they are characterized in a sense whether the donors have parasitemia, the age groups, etc., and this is summarized here in this table. So we have one group of sera that we defined with a very high parasite density. This is a sera from children, so they are exposed to malaria, and the assumed immune status is they are susceptible, so they are because of this high parasitemia. Then we have a second group. These are adults with a low parasitemia, but they are asymptomatic, so they have no disease, so they are assumed to be semi-immune, so they are protected from disease. And just to give you a brief overview, what does it mean, the semi-immunity? So this is also called naturally acquired immunity, and this is what develops slowly over several years through repeated exposure, and this results in a kind of immunity against severe and also to some extent to non-severe clinical disease. But as I mentioned also in the beginning, this semi-immunity or naturally acquired immunity is never sterile. And you could just define the semi-immunity or clinical immunity or describing that as the absence of disease but in the presence of a Plasmodium infection. So these are the 2 mean malaria exposed groups, and as I mentioned also in the beginning, we have a group of malaria naive individuals which were never exposed to malaria, and therefore of course the assumed immune status to malaria is malaria naive. So now starting with the results. So this is just as an example how our stainings look like in these different groups that I presented in the previous slide. So we have here the malaria naive individuals, we have malaria exposed, the assumed susceptible group with the higher parasitemia, and we have the malaria exposed individuals which are assumed to have acquired semi-immunity. And what you can really nicely see is that there is an overall, of course, as we expected, a difference in the overall number of spots, especially here in this semi-immune individual. And there is a clear difference between the semi-immune individual compared to the susceptible and, of course, to the malaria-naive individual. Here you can see how many serum samples we have already analyzed so far, but since the study is still running, this number will be further increased. So at the moment we have roughly 20 sera per individual group. And now I would like to show you the cluster analysis in total for this 62 sera. And this cluster analysis is then done based on the staining intensities of the results. So, and this is the first result of this cluster analysis, and what you can see here are from top to down are the sera, And here from left to the right are the 1,104 peptides that we analyzed. And what you can see, I guess that's quite clear, is that the algorithm arrange here the sera, so the clustering of the sera is, you can just see, from my point of view, 3 main groups. So you have here the group with a very low staining intensity, and indeed these are, as expected, the malaria-naive individuals. Then we have 2 more groups, and these are the two exposed groups, actually. I would say, or from my point of view, the middle clustering is a more heterogeneous group regarding the staining intensities. And here sera from the susceptible donors are clustering in a higher number. And here below you see these very intense staining intensities. Here the sera is from the semi-immune donors are nicely clustering. So this looks quite promising. You could also just have a look on the peptide clustering. So we have here, let's say, peptides here in these malaria-exposed individuals which are stained very intensely, and underneath this first 30 hits here are peptide sequences from already described malaria antigens, but also, and this was very nice, also epitope structures derived from our novel antigens. So this looks very promising. When we can also have a closer look only on these 2 malaria-exposed individual groups, just to look only at these 2, and this looks more or less comparable to the previous slide, but just here the naive group is excluded for this cluster analysis. And again, what you would assume, the clustering regarding the sera divides the sera in 2 main groups. So here, this time, the semi-immune sera are clustered here on top, and the susceptible group is clustered here below. And you observe here in this high staining intensity region of the peptides more or less the comparable peptides that we observed in the previous slide. Well, but in order to really quantitatively determine significant differences in epitope recognition between 2 distinct groups, a bias regularity test have to be applied, and we also did this already with this current data we have, and we have been able definitely to validate epitopes from known vaccine candidates in this exposed group, and additionally we were able to identify epitopes derived from our novel antigens. So, and this is now the basis for our ongoing studies, so we will of course validate these data in analyzing more sera and to further validate the data. So just to conclude and have the future perspective where we want to go and where we are. So with the technology from PEPperPRINT that we used, this enabled us to down-select peptide or peptide sequences associated with protection from disease and sterile protection. So in these ongoing studies that I presented now, this will finally result in the description of epitope structures crucial for protection from disease. And in future work, what we are planning to do is to correlate the antibody epitopes involved in protection from disease derived from this study with epitopes involved in sterile immunity. And with this, I would like to thank your for listening, and of course our collaboration partners in this project. So thank you. So open for questions.
Great. Thank you, Dr. Heiss, for this very comprehensive presentation. I want to encourage the audience again to type any questions you may have in the questions pane. We did obtain some questions. Thank you for that. Also because there were some indicators. We will share the slides with you and also post the webinar file on the PEPperPRINT website. So here are some questions that were brought forward. One of them was, does the vaccine include adjuvants?
Yes, it will. Yeah, definitely. Yeah. So it's a subunit vaccine. So it's composed of antigen component, and it will be formulated with an adjuvant.
Okay. And another question regarding the sera that you analyzed, whether the sera were obtained from malaria endemic areas, also from which country, whether that can be disclosed, and then a comment about whether it was a hypo or hyper-endemic area. Yeah, any comments about this, Dr. Heiss?
Yeah, of course. It's actually from Burkina Faso.
Okay.
And this is in collaboration with our collaboration partner here, Thomas Jenisch from the University Hospital Heidelberg. And it's an holo-endemic region where the sera comes from. So there's a high transmission of malaria.
And another presenter asked whether the semi-immune group is contrasted by patient history. Can you say anything about this?
Depends what exactly does it mean.
Yeah, maybe
we have data on of course on the clinical features. So from the time point where the blood was taken. So actually, as I presented, the parasite density and symptoms or not symptoms, and this data we have here, so they are described.
Okay. I also encourage the person who asks the question, if you feel that the question is not fully answered, you can also contact us offline, and
of course,
we can then further discuss your particular case. Another question here, how many different individuals in each group? But I heard there were 3 main groups, right? Or
so far actually it's 20, so it's from the exposed groups. In the 2 we have 22 individuals at the moment per group, but since the project is still running, this number will be increased. I would say at least double. So at the end of the study we will have more than 40 individuals per group.
Okay. Another question, I'm reading it here. It seems that once identified an antigen as reactive, most epitopes from it are recognized by the sera from semi-immune people, aren't they?
Yeah, that's true, but this is this cluster analysis, and we now have to really look at this quantitatively with this regular Spires t-tests, which are really significant different in those two groups. And I could already tell you these are not all of them. Of course, this you will not have, no. So it's a selection of them that show then really significant differences between the susceptible and the semi-immune group.
Okay.
And this will be then further analyzed, and these are the particular focus then later on.
Another question here, which antigens were included as the previously known antigens in the screen?
All right, so we included antigens from the pre-erythrocytic stage and from the blood stages. So we included the circumsporoside protein as being the leading vaccine candidate when it comes to subunit vaccine approaches in malaria, so CSP. We have a protein which is called XP1. We have additionally two, so MSP1 and MSP2 are the other two. So these are the control antigens.
Okay. Here's another question about the epitope mapping. How do we design the control peptides if we don't know if we will have antigen-antibody reaction? In case, guys, this may be a little bit difficult, I also want to introduce Dr. Lisa Steinberg here from PEPperPRINT. She's a panelist. Maybe she can briefly comment on that.
So hello. So the question was how we design control peptides in this study. So the control peptides that were mentioned by Dr. Heiss are HA and flag epitopes or flag tags. So these are mainly quality control peptides. They are not so much used as serum control peptides. What we could include in such a study would, for example, be epitopes from polio vaccines. So in the Western country, everybody is immunized with polio vaccine. Usually, say around 90% of the sera include antibodies that bind to polio epitopes. However, of course, here in this study, we talk about sera that were taken from individuals in Africa. So certainly, we'll have to discuss for optimal positive controls here. But as I said, the HA and the flag tag are used as control, as quality control peptides.
Okay. Here's another question that may be closely related to the previous one. What can you tell us about the antigens you're using for the PEP scanner assays? The antigens that were used. Does that make sense?
What do you mean with PEP? Do you know what it means with PEP scan?
Yeah, I don't know. This is what the attendee typed.
So for the probably the attendee would like to know which antigens were used in this screen. I think the antigens that were used, as Dr. Heiss said, so they are antigens which are known from one side and antigens that were discovered by Malvar, and these antigens were then translated into overlapping peptides.
Yes.
Okay. And another question here, what known Plasmodium epitopes have been recognized by your approach?
For example, I can give one example for the well-described B-cell epitope in CSP, so this we also pulled down with this approach.
Okay. Another question here. So questions are rolling in. Were some experiments with selected peptides carried out in mice?
Sorry, can you just repeat the question?
Were some experiments with selected peptides carried out in mice?
Yeah, we did experimental vaccination studies in mice, yeah.
Okay.
But not yet with those that were identified here in this project, since this is still running, so it's quite new.
And another attendee asks whether you are planning to work on T-cell epitopes as well.
Oh, we already do that, of course, yeah.
Okay. Here's another question. It reads, this strategy is designed to identify relevant epitopes for the blood stage. How do you plan to find epitopes for the liver stage?
Well, so actually exposed individuals are also, of course, exposed to mosquito bites, and you have, of course, also responses against pre-erythrocytic antigens. So this is not only for blood stage antigens. So you have both.
Okay. Great. I'm still scanning here. Okay, here's another question about the peptide arrays. Is the peptide array available on payment or only through collaboration? Lisa, maybe this is one for you.
So, of course, the peptide arrays can be purchased from PEPperPRINT. Here, this is a collaboration project together with Malva company. But I would say most of our projects are just normal customer projects. So the customer can get in contact with us over the website, and then we'll design the project together with the customer, and then the customer can either purchase the arrays from us and can perform the assays in his or lab, or you can also perform the assays here as a service in our company.
Okay. Another question here. If you identified many immunodominant epitopes, how are you going to make the selection?
So of course what you have to do before you include this in vaccine design, of course you have to prove the epitopes, so whether they are functional or not. So you have to do functional studies looking or proving whether these epitopes or these antibodies that are generated against these epitopes are, for example, have blocking properties or neutralization properties. So of course this is not yet clear when you do the identification, so this is then the next step. And then of course you will select the most potent one harboring, for example, blocking activity in the assays that we can do.
Okay.
Mm-hmm.
Here's another question again to the pertaining to the peptide arrays. How was the selection of the peptides for the microarray made? So how were the different peptides selected for the microarray that were printed on the microarray?
So what we always do is that we find the project together with the customer. In this case, we talk together with Dr. Heiss, and then we decide what is the best strategy for her. And she said that she would like to analyze a high number of samples. And then we decided which overlap we are using. In this case, we use an overlap of 7 amino acids. But usually we use the maximum overlap for epitope mapping studies. But here we are really flexible with our platform, and we always try to find the best or the optimal solution for each project. So, so that's also the reason why we always talk with the customer about the project. All projects are different, so to say.
Okay. Yeah, here are more questions rolling in. Another more technical question. How was the array data pre-processed with respect to normalization, batch effect, or rather we should reformulate here. How was the array data pre-processed with respect to normalization, batch effect removal, etc.?
So actually what I guess this is also what Lisa may just add. So what we also did is an interarray control. So we just analyzed the same sera on different array batches, and we have a very good recovery of that detection. So actually the processing was, of course, we have the usual background subtraction from the staining. Of course, then since we have duplicates, we of course have the mean of that duplicates then afterwards, and that's the procedure how we did it.
Okay. Another question here for the peptide arrays. How to consider secondary structures of the protein when designing the peptides?
This is a very interesting question. Yeah, that's true. So actually, of course, with the technology that we use so far, we are able to detect linear epitopes. But there are also new developments also that you can then analyze also conformational epitopes. So I guess here also, I guess Lisa can just add some words on that development, right? So to include kind of
we have just released cyclic peptide arrays, and these cyclic peptides can mimic looped conformation or conformational epitopes. So when conformation epitopes are the main or the aim to detect them, then we would go to cyclic epitopes or cyclic peptides. And of course, not all but most of them. So this really gives an advantage here to use them.
Okay. Here's another interesting question for the vaccine development itself. It says here, how many peptides do you think can be included in the vaccine, and what kind of a construct?
That's kind of a difficult question, how much peptides. So the question is, of course, whether you choose individual peptides or whether you envisage a larger portion of the protein. So we would rather envisage a bigger part of the protein instead of individual peptides. So, but to really comment on the numbers, I think this I cannot say.
Okay. Here's another question. I think I'm going to read two more after that, and then again we will answer all the questions, but I think at some point we just need to conclude this webinar. So this particular question here, how do the results for the previously defined CSP epitopes compare against novel peptides?
Can you just repeat it once?
Okay. How do the results for the previously defined CSP epitopes compare against novel peptides?
So there are, I guess, when I understood it right, the question, so there are, when you just look in the comparison, so they are closely together. Yeah.
Okay. And then here another participant asked, how did you get the attenuated liver stage parasite for comparison?
So this is an in vitro culture.
Okay.
So culturing human hepatocytes and infect them with the attenuated parasite lines.
Okay. Good. And I'll read the last question. This is more regarding the arrays again. Do you provide service for designing and making peptide arrays? Lisa, maybe this is one for you.
Yeah, sure. So of course, we design the arrays. So we just need to know, for example, the antigens that we should translate into overlapping peptides. We either take the antigen sequence or the UniProt IDs, for example, or the customers can send us an Excel sheet with selected peptides, for example. So here we are also flexible. And we can also perform the service. We frequently do that. It of course saves time and manpower if we do the service here, and we have very well trained staff. And as I said, that I would ask everybody who's interested in such a study, get in contact with us, and then we will discuss the project with you, and then we will suggest different options how we can perform such a project. only tell you, okay, this is the main way to do it, but we will give you options and we will find a tailor-made project with you.
Great. So I think I'm going to start wrapping up here. So thank you all for attending this webinar presentation. We also want to thank our speaker, Dr. Kirsten Heiss, and also Dr. Lisa Steinbrück from PEPperPRINT in answering the many questions that you ask. Again, if any questions remain unanswered, we will actually answer them offline. So I'm still seeing that questions are rolling in. We will make the presentation slides and the webinar file available, so each attendee who participated in this webinar will receive the presentation slides. Also, the webinar file will be posted on the PEPperPRINT website. And again, if you have any further questions, please contact the PEPperPRINT team directly. Please check out our website for updates, future events, and also webinar announcements. I will end this webinar now. Thank you all for dialing in, and thanks again to our speaker. Goodbye.
Yeah, thanks you. Yeah, goodbye.
Thank you, goodbye.